Let's review some biosignaling biochemistry. So thinking about things like GPCRs and RTKs and ion channels. And then we'll go through some practice questions, focusing on what would be the effects of various mutations and agonists and things like that on signaling pathways. Because there are literally infinite numbers of potential pathways, I don't think it makes sense to really memorize various ones, but instead, it's important to know the fundamentals of the key types of components, so things like G proteins and RTKs, and crucially, to be able to interpret signaling pathway diagrams. So the only pathway that I would like you to know by heart, basically, is the GS pathway through to PKA, which we'll go through in a minute. So that's going to be the one that we talked about that's typified by our beta adrenergic and glucagon receptor pathways. And then be able to answer questions using provided diagrams. So if something you can figure out just based on reading a diagram, then that's fair game, basically, for questions. So you can actually learn a lot just from looking at diagrams. And so that's why I pay so much attention to teaching you how to interpret these diagrams and going through practice problems about what would happen in the case of various perturbations of the pathway, things that you can figure out just by reading the diagrams themselves. Because this is going to empower you to be able to interpret any diagram that you come across, no matter how complicated it might look. So when you're looking at biosignaling pathways, the key thing is to follow the arrows and look for signs of activation and inhibition. Unfortunately, these are often going to be drawn differently in different pathways, and different people have different styles, and different journals have different styles, and all this stuff. But when you're looking for activation, you often going to see something in green, maybe. You're going to see arrows, you're going to see triangles, plus signs. For inhibition, you might see these kind of sideways T's, X's, minus signs, these crossed out zeros. I don't know what these symbols are called, but all of these could be signs of inhibition, and all of these could be signs of activation. And sometimes then you're just going to have to put a little critical thinking into things. The critical thinking is helped if you know the basics of various pathways and what processes you would expect a hormone to influence in which directions. And so the key ones that I would like you to just know for now, and we'll get more into them in future lessons, but glycolysis, this is the breakdown of glucose into pyruvate. Gluconeogenesis is the making of glucose from non-carbs. So gluco, sugar, glucose, new, genesis, so making of new glucose. Glycogenesis is the making of glycogen from glucose. So remember, glycogen is our storage form of glucose. And so we've got this branching network of these glucose monomers. So these individual glucose units that are linked up into these chains. These branching chains with lots of points at which you can chew off from and eat. And that when we make the glycogen, we call it glycogenesis. And when you go and your bodies break that down, we call it glycogenolysis. So lysis is this breakdown of something, and glycogenolysis is basically the breakdown of glycogen. And you break it down into glucose, and I have this asterisk here because what you're actually going to be breaking down into is going to be glucose 1-phosphate. This can be converted to glucose 6-phosphate, which, as we'll see when we talk about glycolysis, can actually go through glycolysis and be used for energy. And that's what happens in your muscles. So your muscles and your liver are the two main places you store glycogen. Your muscles is going to break them down to glucose 1-phosphate that goes to glucose 6-phosphate that's used for energy. In your liver, that glucose 6-phosphate, your liver has this enzyme called glucose 6-phosphatase that can dephosphorylate the glucose 6-phosphate so you can let out the glucose and let it into your bloodstream. So you don't need to know these details for now. You'll have this diagram. But the key thing is that the muscles and liver are the two places you're going to store glycogen. The muscles break it down and use the energy for themselves. The liver breaks it down and sends out the glucose into the bloodstream. And it's going to do that in response to glucagon. So glucagon is a hormone that's released in response to low blood sugar. Your liver has receptors for glucagon, but your muscles don't. And as we'll talk about, the glucagon is going to signal through these GPCR pathways, the same ones basically that are used by adrenaline, which is also known as epinephrine. And it's going to have the end result of stimulating the increase in the blood glucose. So I like to remember glucagon says all the glucose is gone. It's going to be released and it's going to do things to increase the blood sugar. So it's going to increase glycogenolysis, so break down glycogen. increase gluconeogenesis, so make more glucose. And the liver, remember, is going to be able to ship that out. And then insulin, it's going to say better let some glucose in, so it's released in response to high blood sugar. It's going to trigger signaling through these RTK pathways, so receptor tyrosine kinase pathways. And that's going to increase glycogenesis, so take in the glucose, link it up into form glycogen, and glycolysis. And so break down the glucose and use it for energy and then make things out of it. And so we'll get more into this in future units. But for now, just remember glucagon, glucose is gone, do things to increase blood sugar. Insulin, let glucose in, glucose is high, let's let some into the cells, let's hook it up to form glycogen, and let's use it to make things. The glucagon is going to act through GPCR, and the insulin is going to act through RTK. And I like giving you this broader overview because then it allows us to look at the details of the GPCR pathways and of the RTK pathways with some context. So let's get a little more into the details. And again, you only kind of need to know the G protein coupled receptor pathway from the GPCR through GS to PKA. You don't have to know what PKA does, but just be able to read the diagrams. And then you need to be able to read diagrams of RTK pathways. And so let's look at kind of the main features, as well as then how we can interpret these diagrams and think about perturbations of these pathways. So we're focusing mostly on these G protein coupled receptors and these receptor enzymes, so these receptor tyrosine kinases. But there's also the gated ion channels. So we talked about these when we talked about membrane transport, how we had facilitated transport, where we had these channels that let molecules through along their gradient, along their electrochemical gradient. So it's an energetically favorable direction, but the molecules needed a passage through. The channels allow them a passage through, and they're going to only open up in response to a stimulus. So in our ligand-gated channels, you have some ligand, you have some small molecule that binds to the channel and opens it up to let the ions through. With voltage-gated channels, you have a change in voltage. The change in voltage is then going to open the channel and let ions through. And in the case of our nuclear receptors, we have things like steroid hormones. These are small and nonpolar. They can diffuse freely through the membrane. So that's going to be simple diffusion. They diffuse through the plasma membrane. They diffuse all the way through the nuclear membrane into the nucleus. There, they're going to affect the transcription of various genes to control the transcription and thus the making of the mRNA, which controls the making of the protein, to control the making of various proteins. These nuclear receptors, these steroid hormones, are the only case in which you have the primary messenger actually go into the cells. So let's talk about primary versus secondary messengers. The external ligands are like hormones, they're going to act as primary messengers. So glucagon and epinephrine and insulin, these are all primary messengers. They're not going actually into the cells. They're just binding to the ectodomains, the outer portions of the proteins that are embedded in the membrane, the receptor proteins, causing those proteins to change shape. So remember, that's called a conformational change. And that conformational change is then going to make something happen inside of the cell. So it binds to the portion that's outside of the cells, the ectodomain, and it causes a change in the portion that's inside the cell, the endodomain. And that change is going to do something like set off a signaling cascade or create the production of a secondary or second messenger. The second messenger is going to be an intracellular molecule. These are often going to be small soluble molecules. They're not always soluble. Sometimes they're little molecules that diffuse through the membrane, but they're not going to be proteins. They're not going to be anything big like that. It's not phosphorylation. It's an actual molecule that's produced in response to the signal that was received by the primary messenger. And so basically you have things like cyclic AMP, CAAMP, which we'll talk about in a minute. You can also have phospholipid-based ones like IP3 and DAG. The only one that you're going to be responsible for is the cyclic AMP, the CAMP. And so the key thing is that the primary messenger is going to bind to the extracellular portions of receptor proteins. It's not going to actually go through, except in the case of your steroid hormones, which are kind of weird in that they're hard to kind of classify as primary or secondary. But you have this primary messenger, basically, that's just going to go all the way in. But even with the gated ion channel, You have the primary messenger binding to the channel, but then it's the ions that actually flow through. In the case of our G protein coupled receptor, the primary messenger binds to the G protein coupled receptor. The G protein coupled receptor is then going to change shape, and it's going to cause a series of events to occur inside of the cell. And in response to activating this G protein is the actual enzyme in this pathway that is going to be activated in response to binding to the GPCRs to the actual receptor protein. So the receptor protein is the GPCR. It associates with this G protein. It activates this G protein in response to binding to a ligand. The activated G protein then goes and it causes this pathway to activate. And the classic example of this is going to be the GS to pKa pathway that we look at. And that's going to trigger the production of the second messenger, cyclic AMP, which is then going to amplify the signal and cause downstream events. In the case of our receptor enzymes, so our RTKs are going to be the ones that we're going to be focusing on, our receptor tyrosine kinases. So these are kinases that phosphorylate tyrosine residues. They get activated in response to binding to a ligand, typically. And then that is going to cause them to phosphorylate first themselves to activate themselves, and then also phosphorylate other molecules. And when you have phosphorylation, you have this negative phosphate group that's added to things, added to proteins, and then it's going to cause the proteins to change shape, and that's going to cause them to activate or inactivate. And then they're going to be able to, when they're active, phosphorylate other proteins and cause these massive cascading effects. And these can be kind of organized by scaffolding proteins, which are big proteins with lots of binding sites for various molecules that can hold things together. So let's look at a little more detail at some of the examples. Let's start by looking at our beta adrenergic receptor and our glucagon receptor. Both of these are going to signal through this GS pathway, where the S is telling us that it's stimulating adenylyl cyclase, and adenylyl cyclase is going to produce the second messenger, cyclic AMP, which is then going to activate other proteins such as pKa. pKa is a kinase, so it can set off a signaling cascade. And so the beta adrenergic receptor and the glucagon receptor, these have separate receptor portions, separate GPCRs. But those GPCRs are both going to be associated with the same G protein, basically. And that same G protein then is going to do that downstream activations. Remember that your liver is going to have glucagon receptors, but your muscles don't. But your muscles are going to have these epinephrine receptors, and so is your liver. And so this is going to allow you to trigger the same processes, such as the breakdown of glycogen, in response to both adrenaline, which is the same as epinephrine, or to glucagon, which remember says glucose is gone. We've got low blood glucose, so let's break down some glycogen. So let's go through this pathway, starting with what's a G protein. And so G proteins are really confusing because G proteins, their enzymatic function is to inactivate themselves. And so G proteins are GTPases. They can break down GTP, they can hydrolyze GTP to make GDP. So chop off this last phosphate group. These proteins are considered quote unquote active when they're bound to GTP. When we talk about active, we're saying that they're able to associate with and stimulate other molecules. So in the case of our GS protein, when it's active, it associates with adenylyl cyclase and activates adenylyl cyclase. When it's bound to GDP, it's inactive. It does not associate with adenylyl cyclase. It does not activate adenylyl cyclase. The G protein, it hydrolyzes the GTP to GDP, but it does not make GTP from GDP. So its enzymatic function is inactivate itself. So the G protein can't activate itself. It can only inactivate itself. It's pretty slow at inactivating itself, but it can be sped up with the help of a GAP. And so a GAP is going to be a GTPase activator protein, and it's going to facilitate the GTP hydrolysis, turning the G protein quote unquote off. So GAPs turn G proteins off by giving GTP zap. So you're going to help the G protein hydrolyze the GTP to GDP, inactivating it. Once you have an inactive G protein, now you need to activate it. Remember that the G protein itself can't make GTP. It can only break down GTP. So its catalytic activity is to turn itself off. So we need a way to actually just swap out the GDP that's now holding onto for GTP. And this is harder than it sounds because that GDP is going to be held much, much tighter than the GTP. So it's holding onto its product really, really tightly, and it needs to be made to let go. Once it lets go, it can pick up a GTP. And so GTP is really high concentration, similarly to how ATP is really high concentration. And you can actually convert between like ATP and GTP and stuff, transfer those phosphate groups pretty easily. So we have really high GTP concentrations, but the G protein can't pick it up if GDP is stuck in it. And so we need proteins that are going to help get the GDP out, get it unstuck, so that the G protein can pick up a GTP. The proteins that are going to help us do this are called GEFs, GTP GDP exchange factors. They're going to help turn on the G proteins by getting GDP the F out. So the GEFs get the G proteins activated by getting GDP out. And they cause these shape changes, and you don't need to know all the details of that, but basically you let go of the GDP. Now you can pick up one of the GTP molecules that are freely floating around, and this is going to activate the G protein. Some G proteins are monomeric, such as RAS, and so we'll look at RAS in a minute when we talk about our insulin pathways. But others are going to be heterotrimeric. And so when we talk about G protein coupled receptors, our G proteins are going to be these heterotrimeric proteins. So hetero, different, trimeric, three. So they have three different subunits. G alpha is going to be the one that actually has the enzymatic activity, the one that actually binds to GTP and GDP. And then these other, the beta and the gamma units are going to be helping with some things like regulation. What happens is that this G protein is going to be activated by the G protein coupled receptor. Remember how the proteins that activate the G proteins, we call these GEFs, and they activate them by getting GDP out. So you can bind to a fresh GTP and activate. So you have your ligand bind, your primary messenger bind to the receptor. This is going to cause the receptor to act as a GEF and activate the G protein. The G protein is then going to release the GDP and bind to GTP. Once bound to GTP, now this G alpha subunit is going to dissociate from the beta and gamma subunits. And it's going to have these kind of lipid portions, these hydrophobic portions that allow it to kind of diffuse through the membrane to find adenylyl cyclase. And so now we have this G protein, just G alpha protein, in its bound state, GTP bound state. So it's active. It binds to adenylyl cyclase, and it's going to activate adenylyl cyclase. Adenylyl cyclase is then going to convert ATP to cyclic AMP, the CAMP. And remember, that was going to be a second messenger. We have this small soluble molecule that is then going to diffuse through the cytoplasm and activate various proteins. The beta and gamma subunits are also going to play important roles that we're not going to talk about. Eventually, what's going to happen is that this G protein is going to inactivate itself. So remember that the catalytic activity of a G protein is to hydrolyze GTP. And so it's going to break off that lost phosphate group, gets it back into the inactive state, the GDP bound state. And now it's going to require the binding of another molecule of our primary messenger, such as, in this case, our adrenaline, or remember that's also epinephrine. That's going to stimulate the GDP to get out so that you can bind a GTP and activate this G protein. One of the molecules that cyclic AMP activates is going to be pKa, protein kinase A. Protein kinase A is then going to be able to do things like phosphorylate B kinase, which phosphorylates glycogen phosphorylase, which then leads to the breakdown of glycogen in your liver. That's going to then lead to the release of glucose into your bloodstream. And in your muscles, that's going to lead to the breakdown of the glucose molecules or glucose 6-phosphate molecules into pyruvate, giving you energy and allowing you to go and run away from the bear and stuff in response to the adrenaline. And so we'll get more into these pathways, but for now, you should be able to read a diagram like this. We see these yellow symbols. These are indicating activation. And we see up arrows, that's indicating the concentration of something is increasing. If we were to see down arrows with the concentration, that would say the concentration was decreasing. And then we have kind of the processes that are happening in each of these cases in both the liver and in the muscles. And we can see that from those one molecule of our primary messenger, we're able to lead to a massive effect. So we have one molecule of glucagon in our hepatocytes or liver cells that can release 10,000 molecules of glucose into the bloodstream. And so we got this massive signal amplification, and all of this is taking place through biosignaling. That was just the GS pathway. There are also other pathways associated with G proteins and associated with different GPCRs and different things like that. Some of these are going to involve different secondary messengers like IP3 and DAG. You are not responsible for knowing this pathway. And so we'll focus on the GS pathway for the purposes of this class. But hopefully you're starting to get familiar with diagrams like this and being able to interpret them. Okay, so let's go ahead and talk about our next class of these signal transducers, these receptor tyrosine kinases or RTKs. And so our classic example is going to be our insulin receptor, but we also see a lot of growth factor receptors. RTKs act as dimers, but not all of them are constitutive dimers. When we talk about constitutive, that basically means all the time. And so this is saying that all the time, some of them are dimers, but some of these are only dimers in response to binding to a ligand. So our insulin receptor is a case of a constitutive dimer. So it's always in this dimer form. It's just not activated. Whereas others of these are in their monomeric form, and then they need to dimerize. Two copies need to come together to form a dimer. And they'll typically do that in response to binding to a ligand. In the case of our insulin receptor, it's already a dimer. It's always a dimer, so it's constitutively a dimer, but it's not active until it binds to the ligand. When it binds to the ligand, so in this case insulin, that then causes the different dimers to change conformations and phosphorylate each other. So this is like autophosphorylation, it's cross autophosphorylation. So these different monomers are phosphorylating each other, and that's going to be activating them. So the key thing is just that you know that insulin is going to activate the insulin receptor and cause the kinase activity to be active. So now you have this active kinase, and we want to think about what happens now. You don't need to memorize this pathway, but you should be familiar with answering questions about this pathway. And you should be ready to answer questions regarding similar pathways. What are the key things we want to look for? We want to look for what's connected to what. We want to look for arrows. We want to look for signs of phosphate being added or removed. We want to look for what's going where. And so let's go and let's look through this. We have our insulin receptor, this INSR. We have insulin bind. That's going to cause the insulin receptor to activate. It was already a dimer, but now it's going to become active as a kinase. It's going to phosphorylate itself, and now it's going to be able to phosphorylate other things. The first thing it's going to phosphorylate in this diagram is our IRS1. IRS1 is this protein that's going to get phosphorylated by our insulin receptor. We can see these arrows indicating that the IRS1 is going to be unphosphorylated. It interacts with the INSR, and now it's phosphorylated. Now that it's phosphorylated, it's going to bind through GRB2. Note here that it's directly showing us that it's binding through that phosphate group. So this phosphate group is acting as a sort of handle that's allowing these to associate. So these wouldn't associate if IRS1 was not phosphorylated, and IRS1 would not be phosphorylated if the insulin receptor was not active. And the insulin receptor wouldn't be active unless insulin was present. Now we have this GRB2 is associating with SOS, and SOS is associating with RAS. You can see this little squiggle sign that indicates it's got some sort of anchor into the membrane, some nonpolar anchor. And we can see that it's letting go of GDP and it's binding to GTP. This is showing us that this is a G protein. Remember the G proteins, it can hydrolyze GTP, but it can't make GTP. And so it needs to let go of GDP in order to pick up a GTP. And the proteins that help it do that are called GEFs. GEFs say, get the F out to the GDP so you can pick up a GTP. So we can see that SOS is going to be acting as a GEF, and then RAS is our G protein. SOS is stimulating RAS to let go of GDP, pick up a GTP. This is going to activate our RAS, and our RAS is now bound to this protein RAF. So now let's look and see what RAF is doing. So we see that we have this curved arrow coming and touching RAF, and it's going from MEC to MEC with phosphate groups. This is indicating that RAF is adding these phosphate groups onto MEC. So it's phosphorylating MEK. And so what type of protein must this be? RAF must be a kinase. So we saw here we had phosphate groups being added by our insulin receptor, and that was the kinase. And here we have another kinase. This one's adding phosphate groups to MEK. And so now let's think about MEK. Now MEK is going to be having a curved arrow going from ERK to phosphorylated ERK. So once again, we see that we have another kinase. We're adding phosphate groups. But we can see that these kinases have different substrates. Different kinases are going to phosphorylate different molecules. And so MEK is going to phosphorylate ERK, and now ERK is going to be able to come into the nucleus. What's it doing? Well, we can see that it's interacting with this ELK1, and this ELK1, it looks like it's getting phosphorylated by ERK. And we can see that in the phosphorylated form, this is indicating that it binds to the DNA and causes transcription, which can then lead to translation, the making of proteins from that gene. So just by looking at these arrows and looking at the phosphate groups and things like this, we can tell what's going on in the pathway. Insulin also acts through other pathways, which is, again, kind of going back to why I don't think it makes that much sense to try to memorize these pathways, since you can interpret the diagrams and there's going to be all sorts of different pathways that you might come across, and there's all endless variations of them. And so it's hard to even find a diagram of a signaling pathway to put in slides and stuff because there's so many, and they're often going to be kind of tailored to the specific one that these authors were investigating. So let's go look at a second type. This one is going to be through PKB. So PKB is also known as AKT. And I know it's really confusing. There's a lot of abbreviations. Before we saw that IRS1 could get phosphorylated by the insulin receptor, and that allowed it to associate with GRB2. Phosphorylation of IRS1 can also allow IRS1 to associate with PI3K. When you see a K, it's a pretty good chance that it's a kinase, and so we likely have another kinase here. And this other kinase is PI3K. It's actually going to phosphorylate this PIP2 to give you PIP3. It would be hard to know that just based on this diagram because it's not very clear, but you can kind of see that PIP2 and PIP3, the PI3K is phosphorylating the PIP2 to give you PIP3. And that's going to allow this PIP3 to associate with PKB. And so I wouldn't expect you to be able to realize that PI3K was phosphorylating PIP2. But you should be able to see that PKB, what it's doing is it's going to be phosphorylating GSK3. So we can see we've got one of these curved arrows. Often what we see is when we have phosphorylation, it's going to be activating a protein. But in this case, we can see that we have this GSK3 that's active, and now it's getting phosphorylated by PKB, and that's going to be inactivating it. And this inactivation, so GSK3, now we get some more complicated signaling pathways because this GSK3, it is going to phosphorylate glycogen synthase. And glycogen synthase, when we see synthase, is like making. And so glycogen synthase makes glycogen. GSK3 inactivates glycogen synthase. If you're inactivating glycogen synthase, you're not making glycogen. And so, but if you inactivate the thing that inactivates the glycogen synthase, then you are going to be making glycogen. So now you kind of get your head all tripped up. And so you want to think back to what's the bigger picture? What is insulin doing? So insulin, remember, insulin says let glucose in. There's high blood glucose. And so we want to let some in and hook it up into glycogen. If we want to be hooking up into glycogen, we want to be doing glycogenesis. So we want to be making glycogen. So we do not want to be inactivating our glycogen synthase. GSK3 inactivates glycogen synthase. So we need to inactivate GSK3. And we can inactivate GSK3 by phosphorylating it with PKB. So I'm not going to try to ask you too quickly questions, but just get used to thinking through some of these situations and the complexity of this, but how you can reason your way through things. We can also see that PKB is going to be sort of acting through RAB and RAC1 in order to bring these GLUT4 transporters, these glucose transporters, to the surface of the membrane. And so we can see it's stimulating this arrow that's showing us that we have these vesicles, which these glucose transporters in these vesicles. And then this is going to allow these vesicles to be brought to the membrane. And so again, I want to ask you something so tricky where you had to realize that these vesicles are being brought to the membrane. But you should be able to see that this is a glucose transporter that's transporting glucose into the cells. So you can see that in response to that insulin binding, through this pathway, we're going to be activating the transcription of genes for cell division. We've got a lot of energy, so let's go and build stuff. Let's make more cells. And here we're also going to be inactivating our breakdown of glycogen, and we're going to be bringing these glucose transporters to the surface of cells. All of this is going to have the end result of lowering our blood glucose. So if we just think of insulin, let the glucose in. If we've got a lot of glucose, let's go, let's use it, let's build things, let's make glycogen, let's stop breaking down glycogen, let's do some anabolism, all these various things. And then think through, does my answer to this pathway make sense given what I know about the physiological functions of insulin, given what I know about the physiological functions of glycogen, about glucagon, all these various things. So let's now get into some practice problems and see the types of questions that I might ask. Starting with connecting our types of membrane transport to our biosignaling. And so let's think about which forms of membrane transport are involved in each of the types of biosignaling that we discussed. So our options are going to be simple diffusion, facilitated diffusion, primary active transport, secondary active transport, or none. Let's think about our GPCR signaling. In this case, we don't have nothing's going through the membrane. I mean, there's a protein going through the membrane, but there's no molecules actually moving through the membrane. Same case with our RTK signaling. But what about our gated ion channels? That's going to be facilitated diffusion. And again, it's not the primary messenger that's going through, but it's the ions. And the ions are being let through in response to the primary messenger or in response to a voltage change. The only case you're actually going to have the primary messenger come in is going to be the steroid hormones, and that's going to be just through simple diffusion. You should be able to predict the effects of agonists, antagonists, and mutations on biosignaling pathways. So let's go through these terms. Agonists, remember, these are going to turn on a pathway, mimicking a natural ligand. Whereas antagonists are going to turn off pathways, such as by blocking the binding of a natural ligand. What about mutations? So here it's really important to think about where the mutations are occurring. And note that I'm going to use this term mutation loosely. So at a technically speaking level, mutations happen at the level of the DNA, and they may or may not cause changes in the protein. We're going to use the term loosely and talk about mutated proteins, which is going to make some people not happy, but it's basically commonplace that we talk about mutations in proteins. So think about if we had mutation in a receptor. We need to think about not only what protein is the mutation in, but where. So if you have a mutation that's going to prevent the ligand binding, or a mutation that prevent binding of intracellular proteins to that receptor, that's going to decrease your signaling or your activity. Mutations that cause overexpression, so you're making excess protein, and/or that lead to constitutive activation, so they're active all the time, even without needing ligand binding, those are going to lead to increased signaling, increased activity. Mutations in G proteins. Here you really got to think through things. Remember that the G protein, its catalytic activity was to inactivate itself. So it could hydrolyze the GTP to GDP, turning itself off. But it can't turn itself on. And so a mutation that affects the catalytic activity of a G protein is going to prevent it from turning itself off. It's going to prevent it from hydrolyzing the GTP, and this is going to lead to increased signaling activity. Whereas a mutation that affects the ability of a G protein to interact with the protein that they stimulate or the protein that activates them, so remember that would be like a GEF, that's going to lead to decreased activity. So if you mutate the G protein in the way that it can't turn itself off, it's going to be stuck on. You're going to have increased signaling. But if you make a mutation that makes it so that it can't associate with the thing it needs to activate it, or it can't associate with the thing it activates, then you're going to have decreased activity. Mutations in catalytic residues of kinases are going to prevent those kinases from being able to phosphorylate substrates, and this can shut down a whole phosphorylation cascade because it's going to affect everything below it. So mutation is going to affect like everything below it in the pathway. And when we talk about a catalytic residue, remember this is going to be one that's directly like involved in the enzymatic function. So changes upstream, earlier, higher up, however you want to think about it in a pathway, are typically going to have wider reaching response, wider reaching consequences than changes downstream, later or later, lower down in a pathway, because upstream changes have the potential to impact more processes. Changes downstreams in a pathway, however, can overcome or cancel out upstream changes, but they can only affect processes downstream of themselves. And so these are just kind of a few different kind of worked out situations if you want to just pause the video and look at those or look at the outline. But let's go through some actual examples. So in our first example, we have a mutation occurring in a catalytic residue of GS alpha. Remember GS alpha, that's going to be the G protein alpha subunit that stimulates adenylyl cyclase to make cyclic AMP, one of our secondary messengers. We have a mutation in the catalytic residue. So that's saying that it's implying that we're abolishing or very much diminishing the ability of the G protein to hydrolyze GTP. Remember that the hydrolysis of GTP was turning the G protein off. And so we're getting rid of the ability to turn it off. So it's going to be stuck on. If it's going to be stuck on, everything downstream of it is going to be stuck on. And so what if we look at the downstream processes? We want to see what's happening to glycogen breakdown. We can see that this pathway is stimulating the glycogen breakdown. And so if our pathway is stuck on, we're going to have more glycogen breakdown. So we're going to have an increase in our glycogen breakdown. We're going to increase our blood glucose. Now let's think about another G protein. This one is going to be a monomeric G protein, and instead of us being associated with a G protein coupled receptor, it's actually associated with RTK. Now we're talking about this RAS protein. Again, we have a mutation in a catalytic residue of a G protein, so it's going to be stuck on. And we want to think about now what's going to be downstream of this, because things that are downstream of our mutation are going to be affected. And so how is MEK affected by RAS? Let's see, RAS is going to be activating RAF. RAF is a kinase. We can see it's adding phosphate groups to MEK. If this is stuck on, RAF is going to be stuck on. And if RAF is stuck on, we're going to be keeping phosphorylating MEK. So MEK phosphorylation should be increasing. What about ERK? We can see from the diagram that MEK is phosphorylating ERK. And so if MEK is stuck on, because RAF is stuck on, because RAS is stuck on, then ERK is going to be phosphorylated more. And finally, what about if we now think a step further, we want to think about what would be the effect on the transcription of cell division genes. Well, ERK, its function was to come and phosphorylate ELK so that ELK could then increase transcription of cell division genes. So if ERK is stuck on, ELK's going to be stuck on, which means that we're going to get more transcription. But now let's think about how this could be influenced by other scenarios, if in the context of having that RAS mutation. And so we would say, okay, we have this RAS mutation. In the absence of any other thing, we would see that we would increase transcription. But how could we get it to decrease transcription, given that we have a RAS mutation? Our options are using a phosphatase to dephosphorylate MEK, so removing these phosphate groups, using a phosphatase to dephosphorylate ERK, removing these phosphate groups, decreasing our insulin concentrations, or stopping ERK transcription. So all of these except for decreasing insulin concentrations. If we dephosphorylate MEK or if we dephosphorylate ERK, that's going to then allow us to have ERK be dephosphorylated so we're not getting the transcription. It'd be very inefficient because we'd keep phosphorylating it and then dephosphorylating and phosphorylating and dephosphorylating it, but we would be able to do that. If we just stopped ERK transcription directly, that would definitely do it. But what about the insulin? Why wouldn't this do it? This wouldn't do it because this is upstream in the pathway. So we could remove insulin. This wouldn't be activated, but this RAS protein is stuck on. And if this is stuck on, then everything down here is going to be stuck on. So we can do whatever we want up here, and it's not going to really impact things down here. So the downstream is going to take precedent, which is why we can do things downstream of the RAS in order to influence things downstream of that. But we can't change things upstream in order to change things downstream of a mutation or something like this. What about if we didn't have this RAS mutation, however? Well, now all of these would decrease the cell gene just transcription. Because if we have less signaling, we're going to have less RAS on, and then we're going to have less of this. But if RAS is stuck on, then it doesn't matter how much insulin we have. And this is why it's so important that these proteins stay non-mutated and why mutations in them are often associated with various cancers. What about the effects of an INSR antagonist on the transcription of cell division genes? So remember, an antagonist is going to prevent the activation. And if we're preventing the activation of the insulin receptor, and the insulin receptor was leading to the transcription of cell division genes, we're preventing the transcription of the cell division genes, so that's going to go down. What if we had that antagonist, but now we had a mutation that caused RAS to be stuck on? Would this be able to overcome that antagonist? Yes, because RAS is downstream. So even though we are blocking this, RAS doesn't really need it. It's become non-reliant on it. It's stuck on, and so you can still have signaling downstream. Few more questions? Which components of the beta-adrenergic coupled GS pathway are these following things? Our primary messengers. So that would be adrenaline. And we can also have glucagon, but in this case, we have beta-adrenergic coupled, so we're adrenaline. And remember, another word for that is just epinephrine. Did we have any secondary messengers? Yes, so we had cyclic AMP. And remember, the secondary messenger was that small molecule inside of the cells that's produced to amplify the signal from the primary messenger that was received outside of the cells. The primary messenger is not coming in the case of the GPCR pathway, and the second messenger, though, is getting released in response to that inside of the cells, so that the message can still reach where it needs to go in the cells. Did we have any kinases in this pathway? Yes, PKA, protein kinase A. What about G proteins? Yes, we had our GS protein was going to be our G protein. So GTP and GDP are not G proteins. These are just small molecules that are bound by the G protein. GS is going to be the G protein itself. There are other types of G proteins, and so there's like GQ and GI and stuff like this in terms of these heterotrimeric ones. And then there are other G proteins, but in this pathway, the G protein was the GS. Let's do the same sort of thing for this insulin pathway that we looked about, where insulin was signaling through this RAS, RAF, MEK, ERK pathway. And so our primary messenger here, this is going to be, remember, the primary messenger is going to be the hormone or whatever, the ligand that binds to the receptor. This case is going to be our insulin. Do we have any secondary messengers? Not in this pathway, no. We only see phosphorylation, and the phosphorylation is not a second messenger. It's a way that you can relay the signal, but a phosphorylation cascade is kind of an alternative to a second messenger in a way. It's not a second messenger. What about kinases? We saw quite a few kinases here. So we have our INSR, so our insulin receptor. That was a kinase. We also saw we had RAF1, we had MEK, and we had ERK. So if you want to see if something's a kinase, look to the arrows and see if it's adding phosphate groups to things. And also a good sign is that you've got a K, but not always. So like RAF1, you don't see a K in its abbreviation, but often you will see a K. What about G proteins? So our receptor in this case was not associating with the G protein, but we did have this G protein down here, this RAS protein. We could see that it's releasing GDP and binding to GTP. And it's doing so stimulated by the GEF, SOS. So now let's look at some more questions. Looking at that same pathway. We have a mutation occur in a regulatory region of the gene for INSR. So the gene for our insulin receptor, we have a mutation that causes this protein to be overexpressed. So we're making more of this protein than we would normally have. What would the effect be on cell division gene transcription compared to the wild type or the normal cells? And we're given the information that this pathway leads to the transcription of genes related to anabolism and cell division, promoted by the phosphorylation of transcription factors that are phosphorylated by ERK. Okay, so we've got a mutation. We're making more of this. And this is going to lead to this activation of this pathway that leads to the transcription of these cell division genes. So if we're overexpressing, we're making more of this insulin receptor protein, we're going to have more activity. So we're going to increase. Now we have a mutation in a catalytic residue of INSR, disrupting its catalytic activity. So remember what type of protein was INSR? It was going to be a kinase. We can see it's phosphorylating IRS1, and we know it's a receptor tyrosine kinase. If we have a mutation that disrupts its ability to phosphorylate things, we would expect that this pathway is going to have decreased activity. What about RAS? We already talked about RAS and how if we had a mutation in a catalytic residue of RAS, it's going to be stuck on, so we would increase transcription. What about a catalytic residue of RAF? So RAF, this is a kinase. If its catalytic function is abolished, it can't phosphorylate MEK, which means that we're going to cut off the pathway. We can't get the increased transcription, so we'd expect there to be decreased gene transcription. And now let's think about if all four of these mutations occurred at the same time simultaneously, what would be the effect? So we have an insulin receptors being overexpressed, but then we have a mutation that makes it so it can't do its phosphorylation. And then we have a mutation that makes this stuck on, and a mutation that makes this stuck off. The key is to look at the most downstream thing, and that's going to be this mutation in the RAF1. So RAF1 is going to be dictating what happens downstream of it. And so RAF1 was decreasing the transcription. So overall, we would expect there to be decreased gene transcription of those cell division genes. And that is how you would approach these questions like this. So look at the most downstream thing and see what its effects are, but things upstream can matter, especially when you have branching pathways. So based only on this diagram, which you predict to have a larger effect on cellular activity, a mutation in PKB, remember that's also known as AKT, or one in GSK3. So remember that this is part of that pathway we were talking about with the insulin acting through this pathway. But we don't even need to know that. We just need to see we have a diagram. We need to be able to follow some arrows. And so we want to think about we have a mutation in PKB. And we can see that PKB is acting as a kinase. It's adding a phosphate group. And it's also going to be stimulating this pathway over here. So we have it kind of changing two pathways. We're influencing two pathways. We're influencing two processes. The phosphorylation of GSK3, which is going to be controlling glycogen synthesis, and the transport of these glucose transporters to the surface. A mutation in PKB, therefore, would influence both of these processes, whereas if we mutated just GSK3, we would influence this, but we wouldn't be influencing this glucose transporter moving. So in this case, we would have that the PKB mutation would have a bigger effect because it's influencing these two different processes. Note that if we had a downstream mutation, this could overcome the effects of this. But right here, we're saying both of these are downstream. And so we're going to influence everything that's downstream of it. And so this is going to then have that bigger influence than if we were just influencing one of the branches. Let's look at interpreting some more diagrams. So what effect does PKB, and remember that's the same as AKT, which we just looked at, what effect does it have on ER gene transcription? So we're going to look at AKT and look at what kind of errors are coming from it and going where. We can see it looks like it's going to be activating ER here by phosphorylating it. But we also see the sign here that it's going to be an inhibition symbol. And this inhibition symbol is going to be the one that we want to look at because it's going to be going directly to this ER gene transcription. So we want to see, the question is asking us about ER gene transcription, and this is going to show us ER gene transcription. We want to look in the diagram and see where that's mentioned. It's mentioned here, and there's an inhibition arrow leading from AKT to it. So we would expect that this would then going to be decreasing the ER gene transcription. And what other receptor did we see activate that same AKT? And that was going to be our insulin. But that was just kind of because we had just talked about it. And I want to ask you to be able to remember that. Going back to integrating membrane transport and biosignaling, are there any depictions of membrane transport in this diagram? And if so, where? And so the only place we actually see something moving through a membrane is going to be through this glucose transporter. So this is going to be an example of our membrane transport, but these are not. Based on this diagram, which hormones activate PP1 protein phosphatase 1? So we have this diagram. We're going to take an overall look at it. We can see we're looking at the breakdown of glycogen. So we've got glycogen, and then we've got glycogen N-1. It's indicating that we are breaking down glycogen, using glycogen phosphorylase to break things down, and glycogen synthase to make it. So we can see that we've got these two pathways with two different enzymes. And so these are different pathways to make it and break it. And these are going to be controlled by this intricate network of enzymes so that we can tightly control the making of glycogen, glycogenesis, and the breakdown of glycogen, glycogenolysis, in response to hormones like glucagon, epinephrine, and insulin, as well as in response to things like our availability of glucose. So based on this diagram, which hormones are going to activate this PP1, this protein phosphatase 1? So we want to look, we see that insulin is going to be activating this IRS1, and this IRS1 is activating PP1. So insulin appears to be activating PP1. Let's see if this makes sense. PP1 is going to be activating glycogen synthase, which is going to be making glycogen. So let's think about the function of insulin. Insulin is saying, let's glucose in, let's link it up into glycogen. So we want to stimulate glycogen synthesis. And so it makes sense that insulin is going to stimulate, it's going to activate the enzyme that's going to activate the synthesis. So sanity check, yes, insulin. What enzymes does PP1 activate? Well, just mentioned it activates glycogen synthase. We can see that we've got this arrow going from it to glycogen synthase. What about it inhibiting? Well, now we want to look for these inhibition symbols. And so we can see that PP1 is going to be inhibiting phosphorylase kinase and glycogen phosphorylase. We also see that these are color-coded in red to make things easier to see, and that we have it in the directionality where we have the inhibition on going from PP1 to glycogen phosphorylase and from PP1 to phosphorylase kinase, as opposed to over here where we see that we have inhibition going from here to the PP1. What would be the effect of a PP1 inhibitor on glycogen breakdown? So now we want to inhibit PP1. Or we don't want to, but we have the question where it wants us to. And so if we inhibit that, what's going to happen? So if we inhibit PP1, we're not going to activate glycogen synthase. If we're not activating glycogen synthase, we're not going to be making glycogen. So we stop making it. But the question is asking us about breakdown. The breakdown was controlled by glycogen phosphorylase. If we have a PP1 inhibitor, well, let's see. PP1 was going to be inhibiting the glycogen phosphorylase. If we uninhibit the glycogen phosphorylase, we're going to be increasing the breakdown of the glycogen. We're increasing the glycogenolysis so we can release more glucose or glucose 1-phosphate. And so let's think about if that makes sense. So let's think about what else inhibits PP1. We can see that PP1 is inhibited by glucagon and epinephrine. We remember that glucagon says glucose is gone, let's break down some glycogen and let some glucose out. So if glucagon is going to promote the glycogenolysis, we would also expect that something else that leads to the PP1 inhibition would promote the glycogenolysis. And so we would increase the glycogen breakdown because we're inhibiting an inhibitor of glycogen phosphorylase, which catalyzes glycogen breakdown. Remember, that's also called glycogenolysis. And so following the arrows, you can really figure out a lot, but then you also need to take your critical thinking cap on and think about whether something makes physiological sense. And that is the key to biosignaling problems. Rather than memorizing things, learn to read diagrams with your critical thinking glasses on. Hope that helped.